How to make color. Here are some beautiful and colorful materials. In human history, we have used many naturally occurring minerals or pigments isolated from animal or plants for a long time. From 19th century, human beings have learned the principle of color generation and started to make synthetic colorful materials. To become colorful materials, the material should absorb certain wavelengths of visible light. Then we can see the leftover colors, in other words, the complementary colors. Then the next question is, how to make such colorful the material? Here is one possible way. You may have heard a particle in a box in quantum mechanics. If there is a particle in an infinite potential well, in classical other mechanics, the particle is moving from one side to the other and bounce back, and it cannot penetrate this well, and it can just moving like this. In quantum mechanics, especially when the particle is very small, like electron, now the electron behaves more like a wave rather than particle. And the electron can occupy one energy state like this, and then it can form another energy state with one node like this. And the electron can jump from one state to the other by absorbing exactly matching energy difference between these two states. And by absorbing another energy, it can jump into the next stage too. And the energy gap in between, it can be provided by electromagnetic wave, and this can match to ultraviolet, and sometimes it can be visible light. Then the next question is, how can you make such a box? The box can contain electron inside, and that energy gap should be matching to the visible light. Then we can make colorful materials. One possible way is to utilize the conjugation. In chemistry, there are two kinds of bond. So one is sigma bond, and the other one is a phi bond. When we have double bond like this, it looks like two equivalent bonds there, but actually it's not. The double bond is composed of one sigma bond and one phi bond. Then this one can act as an interesting model. Let's say the sigma bond acts as a box, and the phi electron is moving around like this as a particle. So this double bond containing molecule can act as particle in a box model. Now, one problem is the energy gap, this one state to the next state, is too high. So they can absorb very short the electromagnetic wave. So it's in the range of ultraviolet. To make the energy gap matching into visible light, we needed to make this box much bigger. Then bigger box has longer wavelengths like this, then it can absorb visible light. So one possible way is bring in another box like this, and then connect this two box with another sigma bond. Then the model becomes like this. The whole box size is like this, and the electron is moving like this. Then the absorbing energy gap becomes lower, then wavelength becomes larger, and chemical structure should be something like this. The double bond is connected by another sigma bond like this. Then the total size of the box becoming double. Now let's see what kind of energy can be observed depending on the conjugation number. If the conjugation is only 1, it can absorb 171 nanometer. Wow, it's too short. The borderline of visible light was 400 nanometer as violet color. The conjugation number is 2. Then it's a 117, still very short. 3 is a 258 nanometer. 4, 296 nanometer. Now you can see some general rule in between, the energy in between of those the conjugations. Still, this is a colorless the molecule. If it becomes a 5, now can you guess what wavelengths of light can be observed? Did you find it? So next one is a 335. It's still quite short, but it may have some broad absorption like this. Now it may start to observe 400 nanometer, and then it starts to show very faint yellow color. How about 6? 364 nanometer. And 7, well, it may not be synthesized yet, or it's not reported yet. 8, it's a 415 nanometer. It's a directly cross the borderline of visible light, and the color is orange color. And 9, not known. 10, well, I couldn't find either. 11, now it's a 470 nanometer. It's red color. 12, not known. 15, it's now 547 nanometer. Now the color change is starting from yellow to orange and red color and violet color. Now it started to cover almost every color in visible light. Let's look at one example molecule, beta-carotene. Beta-carotene is the main component in the carrot, which presented beautiful carrot color. Now we know the color of a carrot, right? So that color is coming from this molecule. And can you count those conjugation number? Let's count together. So here, number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. If the conjugation number was 11, do you remember what was observing light? Well, it was 570, but actually this beta-carotene has 555 nanometer absorptions. Maybe that small change, the small difference is coming from maybe this ring structure or maybe other groups around here. But still the general rule is if you want to make longer wavelengths, you need to make the box bigger and bigger. Now, if it have small part of that conjugation, it may observe shorter wavelengths. Here we see another color wheel. This is a modernized color wheel with the exact measurement of wavelengths for each colors. Now, starting from the 400 nanometer, now this is a violet color, and in the same order of rainbow color, along line, the increasing of those wavelengths, we start to see blue, green, yellow, orange, red color, and our eyes can sense up to about 750 nanometer. So this is exactly the same order of a rainbow. That's additive colors. But the color we see from colorful material, it's not additive colors. That's the subtractive colors. That means if the material observes the light around here, then we can sense the color as yellow color like this. This is the complementary color, you remember? So let's say if you make those conjugation longer and longer, in the beginning, the conjugation was very short and they can observe only UV light. Then our eyes cannot sense any colors. When the material has longer and longer conjugation, if they enter this violet range, they observe the light around here, then we start to see yellow color. Now, let's think about the old books. The paper in the books, they have a lignin material. The lignin has a conjugations. In the beginning, the lignin has a short the conjugation system. That's why we cannot see the color. So the paper looks like white. But over the time, the lignin can be oxidized and it can form longer and longer conjugation, and they start to reach this area, and then they start to absorb the light to this direction. Then in the beginning, the paper to our eyes, it looks like a yellowish color. And then if the conjugation is longer and longer, it enters into orange color. And if it is a very, very old book, now it becomes like a dark red or the brownish color moving to these directions. That's how we see those color changes along line, the conjugation of the length increase. So one possible way to make a different color is we can make this kind of a particle in a box and then make it longer and longer. Then we can, in principle, we can cover all the colors. And it defines the size of those box. Then we can control the colors. One of the problem is if the conjugation is too long, there are too many double bonds in the middle, and any of them can be attacked by other oxidant or reductant, then the whole conjugation is broken, then it loses colors. Actually, this is the principle for the bleach, which removes the colors from our other clothes. In the case, either oxidant or reductant can attack any of those double bonds, then it can remove the colors. That's good for removing color. But if you want to make very strong and stable colors, actually long the conjugation system is difficult to make and also chemically unstable. Then you need to use a different strategy. Now here is one trick. Not only making those conjugation longer and longer, we can add electron donating group here. Then practically this part is increasing those energy state, and the particle in a box, the size of the box is becoming practically much bigger than before. If you add another motif, here is electron donating group and here is electron accepting group because this is electron deficient, then the particle of the box of the shape, this side is energy is increased and this side is energy decreased. The practical size of box is becoming bigger, and also there is a strong electron dipole moment forms, and this electron the dipole moment can generate the power of observing a lot more light, and this is the main principle of modern dye synthesis. So here is one example of a modern dyes. As you can see here, one side is electron donating group, the other side is electron accepting group, and then those system is connected to these conjugation systems. Utilizing this structure and with a different motif around here, we can generate broad range of beautiful and very stable dyes. One example of those dyes is so-called crystal violet, almost the same structure, right? Electron donating group and electron withdrawing group with some other groups around here. It can adjust those colors. And this dye has been used to stain bacteria. And bacteria has two different kinds. One is a gram-positive bacteria and the other is a gram-negative. This strong color here, this purple color is from crystal violet. And depending on those pattern of those staining, if the bacteria is stained strongly with the crystal violet, we call this is gram-positive. And then the pink one here, if it is not stained by crystal violet, we call it gram-negative bacteria. Much later, we realized why gram-positive bacteria is stained better by crystal violet and why gram-negative the bacteria is not stained, because they have a different cell wall structure. But that has been discovered much, much later. In the beginning, when this kind of a classification was made, by the way, this Gram is a person's name. Dr. Graham tested this dye and he found this classification. And this becomes the standard of a classification of all the bacteria. Isn't it funny? So there are many, many bacteria. We didn't know how to classify them. By adding one dye, crystal violet, if they are positively stained, we call positive dye, positive bacteria, gram-positive. If it is not stained, we just call gram-negative bacteria. And it has been standard of classification, the first classification of all the bacteria. It's really funny, right? So this means the discovery of science or development of science is sometimes controlled by available tools. In this case, dye material, the colorful material.